So hello everyone. Thank you, Chairman, for the introduce presentation. Then this. So today I represent a study that has been conducted in our lab entitled An Experimental Approach to Measure Particle Deposition in Large Circular Ventilation Duct. I should say that different institutions are associated to this presentation. So I just briefly give you some list of institutions that participated to this project, mainly located, in our case, nearby Paris or in Brittany, in Nantes. So this work, the collaboration, was done in the frame of a larger project founded by the French National Research Agency, ANR, and it was a 4-year project where we studied actually issues related to indoor air qualities and more specifically pollution transfer in indoor environment. So this is one of the research topic that are conducted in the CERTES lab, place where I work, which is a research center in thermal engineering, environment, and systems. So, as we know, the human health could be associated to indoor air quality and human health, and indoor air quality has an effect on human health. And in this case, in the case of the project, which is called Clean Air Net. So net for network, and clean air network means the ventilation network. This is something which is very challenging for the industries in general, and more specifically for food companies, for example. That was the case of this project. Then we observed that the indoor air quality is controlled by the manufacturers, but very poor attention was paid to the HVAC systems. Then HVAC systems, they play a role in pollution transfer as soon as we consider air recirculation, air handling units, design, and also eventually the resuspension processes which occur when the particles could pass through these systems. Then there's a need, I would say, a real need for direct application and to better understand and eventually, of course, control the pollution transfer, and more dramatically for bioaerosol particles in a food company. So that was the goal of the Clean Air Net project, to study the dynamics of airflows in air handling units in relation with the deposition mechanisms of particles on the surface. And also, the great challenge of this 4-year project was to propose an alternative way of concentrating pollution and eventually developing a design to trap particles. So, yeah, I should maybe mention that large companies, together with our R&D institution, were associated with this project. Then, as a first step, I would say that the first work package in the project was to have a situation to get a diagnosis of these different handling units which operate in these companies. Here you have very schematic diagrams of 3 large companies for dairy and food and seafood products, which are sensible to bioaerosol contaminations, actually. And so they are all equipped with filters, and they are all equipped with filters, and they present specific characteristic, like they are large circular ducts, if we compare them to the other ducts that we can find in other buildings, like offices, for example. They present also, would say, physical specificity in terms of physics. They present a physical characteristic that we have to take into account in order to propose, of course, and to study the mechanism in these particular situations. Then they also present, or they can also, we measured actually within the frame of this project, difference of temperature between rooms, between workshops, between handling units and workshop, which could affect dramatically the mechanism for pollution transfer. So surprisingly also, they all have filters, but we measured particles through air collection devices. And so this graph shows the concentration of, in mass, of particles up to 4.5, for example, if I take this point, microgram per cubic meters for particle size, which are similar to bio organism. And these before and after the filters. So it means that probably the filters doesn't play the full role to stop actually the pollution transfer. Then based on this result, we actually analyze more deeply the critical pooling areas within the ducts, and in order to predict the cleaning time, because cleaning time is really something challenging also for manufacturers. Sometimes it really costs a lot of money actually, and sometimes there's also a way to not to clean but to change the whole system, which is sometimes more efficient in terms of cost. So we actually take the different models in the literature which predict what we call the deposition velocity. Deposition velocity means that the particle mass flux to the surface, so the transfer of pollution from the air to the surface of a duct. It takes into account physical parameters, physical mechanisms like Brownian and turbulent diffusion, gravitational settlings, etc. So based on that model, we could, and work has been done to predict the time. Then at the lab scale, to validate this model, to test this model, to assess this model, we had actually used an experimental way of measuring the deposition velocity. Here is a duct. I mean, this diagram shows you a duct with airflow and particles. Here they are yellow because we used actually fluorescent material. So they are particles in the bulk of air. And the deposition velocity could be expressed as the mass deposited on surface divided by the mean concentration of particles in the bulk air. So that's what we actually tried to, and we did in this study. So, and we challenged this experimental way to measure the deposition velocity, but in circular duct by considering different orientation. I will talk a little bit more about that later. So we had the capacity, we had to find a capacity actually to produce, to generate an aerosols. What we call here a VWAG is a brand of, a type of generator which produce monodispersed aerosols. Basically, actually, it's from droplets of liquid. It produces dry residues. And then we developed a taste loop which mimics actually the HVAC system that we found out, based on the physical parameters that I described before. Then this is the monitoring that we had implemented in the duct. So here is a aerosol generator. The aerosol is injected into the loop, and then we measured airflow velocity, particle size in real time, etc. So we also dedicated a special location for measuring the flux on the surface. Then we could get an idea of, or a measurement of the deposition velocity. That is a very important factor for us to get a deposition mass flux on the surface. And so we also equipped the whole duct with specific devices. I won't go into details here, but basically the devices allows us to predict, or to measure, sorry, the mass depending on the orientation. For example, on the mass deposited on the floor, on the ceiling, or the walls, for example. So this is what we did with also fluorescent material, which is very convenient because we could produce an aerosol with a fluorescent residues that we can use after that we can use and we can extract to measure the concentration, related concentration that we get. So we can have an idea of one of the key parameters is the mass. I won't go into the details. So I just give you some findings, main findings. So as I mentioned before, we have worked based on the first diagnosis within these factories for micrometric particles. So here in this table you can see that there's different size, nominal size. This is the size of the particles that were generated in the duct. And here on the right side, on this graph, you can see a typical, maybe you cannot see very clearly, but this is a distribution granules, the distribution of the size of the particle which were generated in the bench, and they present a peak like this and a typical monodisperse profile. So this is very important for us because depending on the size of the particles, predominant mechanism will be very different from one to another. And so this shows, for example, for 1 micrometer particles, that it is possible to generate something very monodispersed for a very long time, like for long runs, up to 3 days, 4 days long. And the sensibility of to detect actually such particles on the surface, the mass could be very low, down to, so as you can see, this is very low for each particle size here. So this is a real advantage of this methodology actually. So here are some other results where I show you, as I mentioned before as well, if it is possible to discriminate actually data depending on the orientation. So here, the graph here, show you for different trials with good replication that it is possible to discriminate. Here you're on the floor and you can see that the deposition velocity is much higher than on the wall. So we could assume that before, of course, but this is demonstrated for large particles. Then it looks that it works well. If we compare that between different diameters, it works well. And as I also said, that there might be an effect of particle size that we see here. For example, here there's 1 order of magnitude, no, not, yeah, almost 1 order of magnitude between particles of 6 micron particles down to 2 micron particles. And this effect is less evident for small particles. So after that, And in order to compare actually this result in this specific case to other results from the literature, we used a very classical parameter, which is a reduction time, which depends on the diameter of the particle. You have the expression here. So the bigger are the particle, the bigger is the reduction time. So it shows also, these are the data that we obtained, it shows good discrimination between surfaces depending on their orientation, either on the floor, on the wall, or eventually, sorry, on the ceiling. The ceiling is less evident, but this was very significant for the particles that we measure during the diagnosis, particle size that we measure during the first phase of the project. So it gives us, as I said, that the possibility actually to compare with one or with very few experiments from the literature. I would say that there's almost only one, which is quite similar to this one, but it shows a good agreement. For us, it's good agreement for large particles and quite a large dispersion of data around particles where the mechanisms are not totally explained by the models. So this shows also that these data, that were produced here would be used to validate the models for large particles and also to help to develop or to complete the model, which are not sufficient today in order to predict something. So this is a very short and precise example of what we did within the frame of a larger project, of course, but to approach indoor air pollution and transfer of particles within indoor environments. So apply to bioaerosols. So through an experimental study. And in perspective, I would say that there's other mechanisms to study, of course, to complete the models. And also within the frame of the project, which is I think 2012, it was the end of the project, of Clean Air Now project. And it enabled also to develop an application. Because of course the demand from the industrials or for the manufacturers of HVAC system was also to have application. So, or to help for designing new equipments, what would be the predominant mechanism for pollution transfer? Okay, my presentation stopped here, but this shows also that in the frame of such a project, different skills, of course, and multidisciplinary skills has to be gathered, either for food, but also for physics, microbiology, designing process, designing and innovation, new equipment for industry. Thank you very much.